Good morning. Welcome back to Biology 1302. Today we're going to talk about Chapter 6, Respiration and Fermentation, Capturing Energy. Again, full disclosure, closed room, closed door, empty room. I'm here by myself. So, plants make their own food, but animals, such as that bluebird and you and me, must eat food, which gives them the energy they need. Most organisms also require oxygen, which is required for cellular respiration. That oxygen, again, is called aerobic respiration. Cells use energy in the food to make ATP. Molecules in the food, like the chicken breast and the broccoli and the baked potato, are used to produce ATP. The energy carrier molecule used to power cells' activities. Without ATP, cells would die. I generally have a question on the exam covering this data that says if you accidentally took a poison that prevented you from making any ATP, what would happen? And the correct answer is you would die. Again, this is the adenine triphosphate. Here's ribose sugar. Here's the adenine, and attached to the ribose sugar, then you see 1, 2, 3 phosphate groups, which is the adenine triphosphate. Glucose molecules, all organisms, from trees to whales to bacteria, use some form of cellular respiration to make ATP. The potential energy stored in glucose here is extracted, and that energy is put into making ATP. The first step is stored energy is released when the bonds are broken. The first step in respiration is this glucose molecule. And now keep in mind, what we're going to talk about all morning today is going to be the use of glucose. Similar steps take place using all of the sugars and proteins and lipids and other things. If you understand this process with using glucose, it's very easily then just transferred to something else. Same type of steps are going to occur. The first thing that happens is that glucose molecule is going to be broken in half. Here's a spot, here's a spot. That's actually going to burn up an ATP at each one of those places. So you're going to start off the respiration process by burning 2 of the items you're going to try to make, and those are ATPs. ATP is essential because it powers nearly every cellular activity that tissues perform. You see this lady, she's inhaling and exhaling, takes ATP to convert that process. For the muscles of the diaphragm to pull down and pull air in, and then those muscles to relax and force air out, takes ATP to do that. Her muscles are working. It takes ATP for those muscles to expand and contract. She's digesting her meal that she had. Again, that process, the digesting of the meal, makes the ATP, so this process and this process can occur. Aerobic cellular respiration is a series of chemical reactions. The overall reaction is C6H12O6, that's glucose, that's a simple sugar, plus oxygen, we breathe it in, yields carbon dioxide gas, that's what we breathe out, H2O, and ATP. The reactants are glucose and oxygen. The products are carbon dioxide, water, and ATP. When we inhale oxygen, it is required for aerobic cellular respiration to occur. Travels to the mitochondria inside our cells. It's inside those mitochondria where this respiration is going to take place. We exhale the carbon dioxide. It is released at the mitochondria during cellular respiration. Our bodies don't have any use for it. So that's why carbon dioxide is considered a waste material. It's something we produce that we don't need, so our body just has a method of getting rid of it. Cellular respiration occurs in 3 main pathways. You have glycolysis, you have the Krebs cycle, and you have the electron transport chain. Glycolysis is the first step. That's where the word glycolysis literally means sugar splitting. This is where a sugar molecule is split. It forms a product called pyruvate. That pyruvate is then transformed into another molecule called acetyl coenzyme A. We'll talk a little bit more about acetyl coenzyme A in a moment. But Some of the energy that is released in the glycolysis step actually creates ATP. Now, there's not a lot of ATP created in glycolysis. There's some, but not much. The primary thing that is made in glycolysis are some molecules that are going to go further into the chain and the pyruvate molecule. In the Krebs cycle, let me see how this is done. Okay. In the Krebs cycle, a derivative of pyruvate is oxidized. That derivative, again, is called acetyl coenzyme A. Carbon dioxide is released, so it's during the Krebs cycle that the carbon dioxide is released. More energy is transferred to electrons. Again, if you look at this little drawing right here, you're going to see ATP being formed right here, but not a whole lot. You're going to see, here's the Krebs cycle. You see ATP going here, and if you look, here's the things that are coming out of, glycolysis. You've got these molecules called NADH. You've got two acetyl coenzyme A's, which are going to go into the Krebs cycle, and then there's my a little CO2 waste material. Now we're going to take these NADHs. They're going to go into the Krebs cycle along with the acetyl coenzyme A. Some, again, you see the arrow pointing that way, so some NADH is being produced. You also see some more CO2 being produced, and you get some ATP. Then all of these products on the left-hand side in blue, the FADH2 and the NADH, are all going to go into the electron transport chain, and at the electron transport chain, the energy in these electrons is going to be slowly removed. Each time some energy is removed, a new ATP gets formed in the electron transport chain. The final receptor at the end of the electron transport chain is oxygen, and at the end of that chain, there's going to be two hydrogen atoms left, and those two hydrogen atoms will connect to the oxygen to form water. So, oxidation reduction reaction, pathways for aerobic respiration release energy by oxidizing glucose and reducing oxygen. And if you remember, oxidizing means we're removing electrons, reducing means we're adding electrons. Since oxygen strongly attracts electrons, the reaction is forced downhill as it releases its energy. Cellular respiration releases energy from glucose, one bond at a time, in a series of many steps. If all of the energy were released at once, most of it would be lost as heat. So by taking a little bit of energy at a time, we reduce the heat loss. Remember, every time we convert energy from way back at the beginning of the semester, every time energy is converted from one source to another, some of that energy is lost as heat. Small steps allow cells to store more energy, more of the energy of the released ATP energy. Where in the cell does it take place? Glycolysis always occurs in the cytosol. So in the fluid inside the cell, here, and in all these little void spaces on the animal cell, and in the little void spaces here in the plant cell, that's where glycolysis takes place. In prokaryotes, the Krebs cycle also takes place in the cytosol. The electron transport chain is found in the cell membrane. For eukaryotic cells, which contain mitochondria, the Krebs cycle and electron transport chain are found in the mitochondria. And this is a mitochondria that's been cut in half, so you can see the cross section. There's another one, and here are the mitochondria in the animal cell. That's a mitochondria, that's a mitochondria. So those little beam-looking structures are the mitochondria. Within the mitochondria, where does certain things take place? Well, if you look at the cross-section of the mitochondria and you see the fluid here and here, that is something called the stroma, and that is where the aerobic cellular respiration is going to take place. The Krebs cycle and the electron transport chain happens inside the mitochondria. The Krebs cycle takes place in this is a blow up of this. This is the inner membrane. This is the outer membrane. So we're seeing this little loop right here. This loop is that loop. This wall is that wall. Some of the reactions take place here. Some of the reactions take place there. So glycolysis is universal. All cells undergo glycolysis. So, on your exam, which of the following statements is true? Glycolysis only occurs in animal cells. Glycolysis only occurs in plant cells. Glycolysis sometimes occurs in bacteria, or glycolysis occurs in all cells. Glycolysis occurs in all cells. It requires many steps, all of which are completed in the cytosol. So it happens, here's the mitochondria. So all of the stuff is out here in the cytosol. Most of the steps require oxygen. So cells can use glycolysis in both cells can use glycolysis in both oxygen and aerobic or oxygen-free environments. I think I just said most. I should have been saying none. In case I did say most, let me make that correction right now. None of the steps of glycolysis require oxygen. It is an anaerobic process. In glycolysis, the glucose gets split. We start with a 6-carbon sugar, and we wind up with 2 3-carbon pyruvates. Reaction converts molecules of ADP into ATP, so we wind up with 2 of those. And then the electron carrier NAD picks up electrons, forming 2 molecules of NAD. So we start here, we have 2 of these. This is my input, 6-carbon sugar. Wind up with 2 NAD, 2 ADP to that to form a net output, 2 3-carbon sugars, 2 NADHs, energized molecules, and 2 ATP molecules. In the transition step, which lots of textbooks leave out, I do not understand why, but it does, the 2 molecules of pyruvate are converted into 2 molecules of acetyl coenzyme A. It is actually acetyl coenzyme A that goes into the Krebs cycle. Pyruvate can't enter it in the pyruvate form. It must be modified, and that happens in the transition step. Each carrier NAD picks up electrons, forming 2 molecules of NADH, and this reaction releases 2 molecules of carbon dioxide. So here again, input, 2 3-carbon structures, 2 NADs. We wind up with 2 NADHs. We wind up with now a 2 3-carbon structure with a little coenzyme A on it, and 2 of the, 2 carbon sugar, and that's where the carbon for the CO2 comes from. You put in 3 carbons, the output is a 2-carbon coenzyme A. Coenzyme A is vitamin A. If you take a nutrition class, when you talk about coenzymes, you refer to them as vitamins. Acetyl coenzyme A molecules are disassembled during the Krebs cycle. Their carbons are released as 2 molecules of CO2. And you'll notice the Krebs cycle takes place inside this membrane right here, within this inner membrane. Again, the input, you have the 2 carbon acetyl coenzyme A's, 2 ADPs, 2 phosphate ions, 6 NAD pluses, and 2 FADs. And the output, there's some more carbon dioxide being formed, there's 2 more ATPs, there's 6 NADHs, and 2 FADH2s. The electron transport chain, the electrons that are bound up in the NADH and FADH2. And again, you can look these up in your textbook if you'd like. NAD is a word, it's nicotinamide, blah, 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 blah. It's about that long. FAD is a word about that long. So we're just going to go FADH2 and NADH. Their electrons are unloaded. Potential energy is used to produce a proton or a hydrogen gradient. That hydrogen ions will then move through the enzyme's ATP synthase, releasing energy. And each time that energy is released, we generate an ATP. And here's the reaction line. You see it's happening within the membrane. Here, the hydrogens are being produced. Here, the hydrogens are being used. ATP is being created. We're getting a bunch of this hydrogen in here. So we've got a high gradient. To make ATP, the hydrogen has to exit. So it exits through diffusion, going from an area of high concentration to an area of low concentration. But you will notice it's taking place within this inner membrane, along the length of the inner membrane. The final electron acceptor is oxygen. Again, here we have oxygen and two hydrogens, which ultimately wind up being water. Water is the final thing that comes out of the electron transport chain. Aerobic respiration yields 36 ATP, and as often is the case, there is some confusion how many is actually produced, and it depends on the semantics that the particular textbook author wants to do. If you will notice, you have 38 here, but you minus 2 for the 2 you used during NADH transport, that very first step that had to happen. You wind up with a grand total of 36, and you will notice in parentheses under 36, it says theoretical yield. That's the number of, I'm gonna get it in a minute. That's the number of ATP that in theory will be formed this way. However, it's not always that number. There's biological entities are notoriously inefficient in how they do things. So while in theory we can get 36 ATPs out of a glucose, probably we don't get that many. So again, that is a theoretical yield. And if you want to know how many ATPs are made during the electron transport chain, 34. So we've got 2 being made during glycolysis, 2 being made during the Krebs cycle, 34 being made, during the electron transport chain, for a grand total of 38. But you burned up 2, that old business thing, you got to spend money to make money, you get a yield of 36. Carbohydrates enter the energy pathways the same way, some of them. You'll notice the green running down the middle. We start with starch and glycogen. Those things are broken down to glucose and glucose-like things, and then the glucose goes through glycolysis, et cetera, et cetera. Fats and proteins go through things a little bit differently. The first thing that happens in protein production, for instance, proteins are made up of long chains of amino acids. The first thing the cell does, the first thing we do for digestion is we take the amine group, represented by the blue dot at the end in the middle. Amine is chemically NH2. That amine group is removed in the cytosol, and you see it exits the cell as the waste product. That's where the ammonia component of your urine comes from. Sometimes, your urine may smell quite, have the smell of ammonia. That's where it's coming from. It's those amine groups getting together and forming NH4. Then again, in the center of the proteins, when you take the amine group off a amino acid, what you are left with is an organic acid. We didn't go through all the gory details of the Krebs cycle, but the Krebs cycle is a series of reactions that takes an acid, citric acid, and converts it. As energy is being used up, citric acid is converted to malic acid is converted to succinic acid is converted to alpha-ketogluteric acid is then converted to a derivative of alpha-ketogluteric acid, and finally that derivative is converted back to citric acid. That's where we come with having this cycle. But what happens in the amino acids after we remove the amine group and we're left with those organic acids, all of those organic acids can enter into one of these phases. Some of them will act like pyruvate and enter through a transition step into the Krebs cycle. Others will simply move directly. They don't need to be converted. Others may enter this cycle at some point along the ring. But ultimately, all of the organic acids from proteins, for protein digestion, are going to wind up going through one or more of these steps. Then fats, we're going to divide those fatty acids. You remember when we talked about chemistry, we talked about the way fats are made, that they're a glycerol head with a fatty acid tail. Well, glycerol is a derivative of glycerin, which is a sugar, so it's going to come in somewhere in the glycolysis stage. The fatty acids, again, you remember I just told you all of these acids. You take the amine group off of an amino acid, you are left with an organic acid. These fatty acids are also organic acids, so they're going to come in here, they're going to be converted, and then they're going to enter the Krebs cycle. And if you'll notice, here's my CO2, my water, all waste products, waste product of nitrogen, and all of those waste products will exit the body. The CO2, you're going to exhale it. You exhale some water. Every time you breathe out, there is water vapor in there. So you're getting rid of some water that way. And also some of that water will go in and mix with the amine groups to form your urine. Again, we talked about the way proteins and fats exit the cell. That's what I just went through. Cellular respiration is not always aerobic. Certain organisms can survive in the absence of oxygen. We refer to those as anaerobic organisms, anaerobes. Glycolysis produces ATP without requiring oxygen. So we start off with ATP, and we start off with glucose. It undergoes glycolysis. It produces a couple of different products. Produces CO2, and or it will produce, in this particular case, alcoholic fermentation will produce alcohol. If you are a connoisseur of what I refer to as adult beverages, beer, wine, all of those things, those are the result of alcoholic fermentation. I had a student several years ago said, "Oh, Mr. Russell, I don't deal with beer and wine. I only like the hard stuff." Well, so if you're a Scotch or a bourbon person, that's fine. You still have to have an alcohol step. Those are also distilled, which is what makes them the hard liquors, but you still have to have you have to have a fermentation process. You have to produce the alcohol somewhere, and that's in the fermentation process. And then the distilling part is removing water, so you're left with a more concentrated alcohol. But again, the result of alcoholic fermentation. Lactic acid fermentation. If you've ever had a muscle cramp, if you've ever been walking on the treadmill, walking up steps, just mowing your yard, and you got a Charlie horse and your muscles all cramped up, those cramps are the result of a buildup of lactic acid. When you're working your muscles, when you're doing those curls with the 40-pound dumbbell, those muscles are working not aerobically, but anaerobically. And if you work out enough to where you are building up lactic acid faster than your body can clear the lactic acid, and you get a lactic acid buildup, you wind up having a muscle cramp. Many prokaryotes and some eukaryotes do this process. Fermentation is what it's referred to. Again, it is also known as anaerobic respiration. We talked about this. The absence of oxygen, cells can use fermentation to form 2 molecules of NAD. The NAD then is used to power the reaction to produce this, and then this is continually producing this, which is continually producing that. You've got the input, 3-carbon sugar, the output's 3-carbon sugar, but you put these in, and the output is the lower energy molecule. Fermentation uses pyruvate to oxidize NADH plus, forming NADH to form NAD plus. The pyruvate is then converted into alcohol, lactic acid, or other byproducts. No acetyl-coenzyme A is made from the pyruvate. Therefore, no acetyl-coenzyme A, no need to have a Krebs cycle. So everything is done through glycolysis. And again, Your textbook talks about champagne production, talks about alcoholic fermentation, also talks about lactic acid fermentation. And I went through both of those. Last couple of things about respiration. In this particular flower, which I'm not going to, philodendron Solomoescens, there we go. These are plants that warm their flowers. They use an alternative pathway. What happens is in their electron transport chain, they prematurely transfer electrons to oxygen, which instead gives them a faster, and what did I say a moment ago would happen if you release the oxygen too quickly? If you released too much energy in that electron transport chain, a lot of it would be lost as heat. This particular plant does that, and by doing that, the flowers actually are warm. The petals and the stamens and stigmas are warm. And what benefit would that be? Why would they need to have why would they need to be warm? Well, the pollinators that are coming in there can actually detect that heat, and therefore they know that that plant is ready to be pollinated. In this case, an inefficient electron transport chain benefits the plant. Beetle pollinators are attracted to the heat. The hot flowers therefore increase the reproductivity success of both the beetles and the plant. By giving off that heat, the beetles can find the plant. Therefore, if the beetles get food, the more they can eat, the more they can reproduce, and the more they wander around inside the flower, the more pollen they pick up, go to the next plant, pollinate those flowers, beneficial to both plant and animal. If you have any questions, please contact me, trussell@uaptc.edu, or you can always send me a message through the contact link course messenger tab. Have a great weekend. See y'all next time.